Women in Science Without Borders initiator, Professor Amal Amin. So today I would like to just give you a brief talk on non-thermal processing of fruits and vegetable. Being a food scientist and technologist working for more than 2 decades at Institute of Chemical Technology, one of the renowned institution in India, and currently I'm also executive committee member of Global Young Academy, and also representing member of Indian National Academy, where we are actively involved and engaged in doing science which can help to the society. So considering that aspect in mind, and here also you would also know that Asia currently, or many countries, the malnourishment is one of the topmost problem, and unfortunately 1 out of 9 people in the world are affected with the malnutrition, and children especially are undernourished, and this is all because of the crisis in the food and nutrition crisis. So it's a very, very heartening fact. We have to accept that in the world, 795 million people in the world are facing food and nutrition crisis. And it is again very, very hard truth to digest that poor nutrition causes 45% deaths in the children which are just below 5-year age. So it's a very, very hard fact. But unfortunately, at the same time, whenever I wonder, being a food technologist, food scientist, and some of you are also working in the area of food science and technology and nutrition, you are environmental scientists, you know that we are not behind in the production. India is one of the second largest producer for fruits and vegetable. China, Brazil is the third largest producer for. So we have plenty of fruits, we have plenty of vegetable, we have plenty of overall food, but only what is lacking is, around more than 40% of the produce goes into the garbage. In fact, I would rather say even more than 80% and sometimes 100%. This COVID-19 pandemic has so adversely affected on the farmers. At least that scenario I have seen in India, that it has adversely affected to the farmers, that even 100% produce has been just gone into the garbage. It's very, very heartening to digest that. At one side kids are dying because of the hunger, and then other side we are throwing out the food. It is also fact that we scientists and technologists, we have been continuously working in developing technologies which are more cost effective, which are environmental friendly, which will be quick, which are very easy to handle. And there are many such technologies that are coming into existence, but those technologies are not very cost effective, they are not even environmental friendly, and initial cost investment in those will not be very affordable to at least the poor countries, the developing countries like India and some other Asian countries. So today I'm going to talk about the technique, or the novel technology that we have been discovering and we have been working for past more than 4 years, and that is nothing but it is a hydrodynamic cavitation technology. So hydrodynamic cavitation is not a new technology, and it has been used for many decades in water. So disinfection of the water or reduction of BOD level of water. So this basically cavitation is a technology when liquid passes from a low pressure area to a high pressure area, the cavitation occurs, and because of the phenomena, which is a natural phenomena, the collapse of the vapor filled cavities accompanied by intense pressure waves, and there is a vigorous shear force, localizing heating events and formation of reactive species and free radicals. And these effects are happening at a millions of fractions of seconds of time and at a millions of locations in the reactor. So we have to have only a small reactor. Actually, this work was also shortlisted recently, falling walls in Berlin's ventures, wherein you have to have a very innovative idea, and I was amongst the top 10 shortlisted candidates who had a very innovative idea. And this work of mine was recently shortlisted last year. I was presenting this under Falling Walls Berlin initiative. So this technology is a very advantageous, because currently whatever fruit juices, see right now there are number of fruit juices that are available or vegetable that are available in the market. uh, so basically, the cost involved in the processing is a very high. At least they are processed by, the most common one is a pasteurization or sterilization, and we all know that what is pasteurization. It is like heating of the juice in such a way that the microbial load is reduced, so that you have that FDA standards to meet upon, like 5 log reduction in the microbial, so that the juice doesn't get spoiled easily because of the microorganism attack. And the second important thing is, like, most of the juices have some of the enzymes, like for example in case of orange you have pectin methyl esterase, and because of the activity of pectin methyl esterase, immediately after quenching or squeezing of the juice becomes, separated, sedimented, and also it becomes bitter in taste. It is because of the action of pectin methyl esterase onto the substrate, that is pectin, naturally present into the juice, and therefore the bitterness, causes the juice, not acceptable. And same is for other juices, like the enzymes PPO present in the apple causes browning, and therefore, it's not very cost effective. So currently the market is the hack. Industries are processing juice by using pasteurization, sterilization, where temperature is beyond 100 degrees Celsius, and we know that these fruit juices are abundant in bioactive constituents, having variety of health promoting effects, be it a cancer protection effect, cholesterol lowering effect. I'm not here to list out all those health benefits provided by fruits and vegetable. It is recommended by many health organizations that we must consume for prevention of cancer at least 2 fruits every day and 1 vegetable every day in our everyday's diet, and there are so many health benefits. So it is always advisable, but then how to process, because processing cost would be too expensive, that right now in the market there are a lot of processed food products juices are available, but they are not with their bioactive constituent, not with the vitamins, not with the and they are added with lots of preservatives, chemicals. And I am not again here to make you aware what chemicals and preservatives and how they would affect on your body, be it causing cancer, be it causing any other adverse effect. So people are becoming, like consumer like you and me are becoming more aware, and we want everything to be natural, more healthy, more in its as is form. Okay, so then when you want those to be as is and more natural form, you have to invent on some technologies which are novel, which are non-thermal, which doesn't produce heat. But then how to preserve them, because then if you do not have heat, right now also, there are many industries, it's like they think that only heat can reduce the entire microbiota or inactivate the enzyme, but there are novel technologies like cavitation, which can at lower temperature inactivate the enzyme. This recently, for 1 year I was on a CNPQ TWAS postdoctoral fellowship in Brazil, and where I worked on various fruit juices, because as I said, Brazil is amongst the third largest producer for fruits, and I there worked on many fruits and vegetable juices which could be inactivated at a lower temperature, retaining highest bioactive constituent, also maintaining the highest sensory analysis as it is similar to your natural one. And not only that, during the storage also they perform so well. So this is like recently I have also published this as a review article, and it is amongst one of the highly cited review paper, because this is the first of its kind review paper especially published in case of application in the food. Actually, cavitation technology and its use in the water is not new. In fact, the cavitation used in water is to prevent. See, generally, you have the water pipings to flow or take the water from one place to another place. So cavitation happens naturally because of the cavitation phenomena. The high pressure and low pressure areas are naturally created in the pipes when the water flows, and then that is undesirable in the piping, so the cavitation causes a lot of pitting, and then the pipes gets corrode. So the cavitation has so much of power that the iron and steel pipes gets corrode. If that power could be consumed or utilized in the right direction, to erode or to kill the microorganism, that is a boon to the technology. And that is what we have exactly done. We have just applied our logic that water having, the fruits and juices, vegetable juices, has more than 90% of the water. And if the same phenomena can be created, only thing is you have to design a reactor so that you can create a cavitation of a different baric pressures. And generally a large amount of energy is released during a pressure of 100 baric to 5000 baric, and a heat of 1000 to 10,000 kilopascals is generated. And these effects are seen at the millions of locations in the reactors, and the bubbles explodes out. And then it is also said that the reactive species like hydroxyl ions and radicals are formed, and then they kill the microorganism there and their effect itself, without causing any loss in the bioactive constituents. So I have already explained how today's consumers have become more conscious about. Yeah. So as I mentioned, this is the oldest and most common application of cavitations. And so therefore we have been exploring this technology for sterilization of liquid foods. We have used this technology more recently in orange juice processing. As I mentioned, in oranges you have this pectin methyl esterase enzyme, which needs to be inactivated. And if you, otherwise, who doesn't want to drink the freshly squeezed orange juice? It's so tasty. Orange juice is one of the topmost juice having highest amount of vitamin C content after amla, but amla is very difficult to consume, and orange juice is the most widely consumed throughout the world, the juice, and then apple is the second one. So it's very important to have this orange juice in its original form. There are a lot of health promoting effects, lots and lots of, it improves the satiety value. There are so many reports wherein it has anti-diabetic property, it has cholesterol lowering property, it protects against cardiovascular diseases. And in my laboratory also we have been working exploring some of the functional properties like antioxidant activity. So it's a very, very beautiful juice. Especially for COVID-19, there are numerous papers found that this vitamin C can prevent this COVID-19 infection. There are reports, published in journals of good standards and repute. So based on that, I have been claiming. So we just did hydrodynamic cavitation of the orange juice at different settings and zones, and we performed some statistical analysis, optimized parameter. We did physicochemical analysis, bioactive analysis for control HTST treated, that is high temperature treated juices, and then cavitated juices at different pressures, time, temperatures. And we realized that it is a wonderful, it is a magical, it protects all the bioactive constituents. It gives you a very, very homogeneous juice. You don't have to use homogenizer, you don't have to use any homogenizer, you don't have to, it saves you a lot of time, cost, and it's so quick, it's so magical. And recently we have published this work in one of the topmost journal, Innovative Food Science and Emerging Technologies, on orange juice. And then we have been working on, because there is a time constraint, and being a moderator I should be on time, so therefore I'm skipping. Those who are interested, they can write back to me. I will post my email ID, and I'm looking forward for more collaboration. We have been also exploring to use this technology for preservation of milk, cow milk, bovine milk, as well as the plant-based milk. So because in the milks there is a lot of separation phenomena, there is enzyme activity, so it's very difficult. Milk is considered as a complete food, and preserving that complete food at a very low cost and also preserving the bioactive constituent is very important. And this again work was published recently in a very high impact factor journal, Ultrasonic Sonochemistry, and this is available online this year. This last month only we published. So generally in the milk, the separation is observed. Here you can see the separation, but because of the cavitation, which is more than 12 days it was preserved, and it looks as it is. And same is for orange juices also. So we have been also we have also worked on peanut, wherein peanut milk could be also preserved. So other fruits that are currently we are working on is tomato juice, sugarcane juice, amla juice. Anything that has a higher water content could be explored, and there are very beautiful and significant outcomes, and we have future plans to explore this technology on other liquid foods which has more moisture content. And I would like to thank all my students who have been working very hard to get these, because initially it becomes very hard to design a reactor and then take trials. Many of our trials and initial years went failure, but my students supported me. We did not got feared of all those hurdles, and challenges did not came into our way. And I'm thankful to Global Young Academy, my institute, SARE by Ministry of Food Processing, and TechWhip, who have been supporting. We have received grants for this project, and I would like to thank you. And I'll be happy to collaborate on a similar line with all the scientists from different countries who are interested onto the similar line. So I will be happy to take any questions if there are. Thank you so much. Yes. Amal? Yeah, so I hope I have finished on time, and thank you everyone. Thank you once again, Amal, for this wonderful opportunity. So I would like to move ahead with our next speaker in the series, and we have our next speaker, Professor Mohammad Kazim Ali.
Oh, thank you so much. Can you hear me?
Yes, I hear you very clearly. Let me give you give your brief introduction, Mohammed, Dr. Mohammed. I have prepared the slides. I would like to show those slides. Yes. Okay, can you just stop this? Okay, no problem. I will not share them slide. Yeah. So, Professor Dr. Mohammad K. Ali, we are so happy that you are here today with your exciting talk on the topic agriculture, biotechnology, and climate resilient healthy rice in Pakistan. Dr. Mohammad Ali is currently working as a professor at KIBG University of Karachi, Pakistan, and he did his MSc from Department of Biotechnology, University of Karachi. Later on, PhD again in biotechnology from KIBG, University of Karachi in 2015. And later on, he went to Kunming, China to pursue again his dream to do postdoctorate in again the similar subject of biotechnology and genetic resource from very known institute, that is Institute of Human Academy of Agriculture Science, that is YAS. And his majorly research interest is agriculture biotechnology, especially genome editing and precision breeding for high yielding and high quality food. Not only this, further recently he has developed his interest more focusing on identifying and finding out new genes, proteins, and regulatory mechanisms, especially for functional food. I'm so happy that Dr. Mohammed, you do also work on functional food. Who should not work? Everybody, functional food is such an important topic because at the same time, increasing malnourishment and diseases across the different country, food can be the boon and food can be the medicine. So Dr. Mohammed has been handling many projects which are funded by governments, international as well as national government organization. He has published numerous research papers in the journals of high repute and standards, and he has delivered many talks. So we are thank you, Dr. Mohammed Ali, for accepting our invitation and being here today with us on this World Forum from Women in Science. Over to you, Dr. Mohammed.
Thank you so much for the introduction, and welcome you all to this webinar. And but today I'm really happy to interact with the scientists just like you and all the participants here. And just before the pandemic, a delegate from the Duhok University visited our university, and we shared our interest with them, and I was very happy when I came to know about this webinar, about this seminar. But no, due to the COVID-19, we could not attend physically in Duke University. I really missed this. My topic is agriculture, biotechnology, and climate resilient healthy rice in Pakistan. As you know, that this session is about the science for humanity. So we really need to think about humanity on the ground reality basis, because you know that technology is advanced in every field of the science, but the human being, especially in the developing countries, are really facing challenges in terms of healthy food. If we consider because rice is an important staple food for half of the world population, and for Asians it is very important part of their diet. So rice, healthy rice is very important, because I will discuss with you that why healthy rice is important for human health. And at the same time, the climate is changing, so there is but we need to balance these two, that we need more food to combat the growing population, but as well as we need a healthy food, because they are associated with the socio-economic activities and the human development. So Pakistan, you know, that is an agrarian country, and hence agriculture development is a prerequisite for achieving food security. So according to the economics of the government, that there are 19.5% to Pakistan share from agriculture. So it is constitutes 42% of the labor force and 65% export earnings, and provides livelihoods to 62% of the population of the country. However, food security is still a key challenge due to high population growth, rapid urbanization, low purchasing power, and many other reasons in this. So according to the food security assessment, the grown reality is that 18% of the population in Pakistan is undernourished, as already Dr. Arya told about that. Malnutrition is really challenging for in this era. So high level of severe stunning say that there are 40 to 45%, and 15% are wasting and underweight, there are 30%. So the malnutrition problem are high in rural areas, because as I told you that in rural areas the people have low purchasing power. They cannot have even enough money to buy vegetables and fruits that are rich in malnutrition, but they're only depending on some crops like rice and wheat, but there is no much in their grain as in nutrition. So in certain regions, like different regions of Pakistan have different percentage of this malnourishment. So similarly, half of the population is consuming less than the dietary requirement of vitamin A and iron. So these are the reason to develop in developing the chronic diseases, which is the disease that called non-communicable diseases like diabetes, obesity, colorectal cancer, and constipation. So according to the National Nutrition Survey in 2018, said Pakistan suffered from high rate of micronutrient deficiency, as I told you before, due to low dietary diversity. Because dietary diversity means they're only dependent only some food, only some crops. There are not much options available as a dietary diversity, affecting mainly children and women. And following the 2010 and 2011 flood, level of acute malnutrition increased significantly. And Pakistan is currently lagging behind in reducing malnutrition, and so many challenges we have. So in addition to hidden hunger, the double burden is coexistence of undernutrition, which means that overweight and obesity and diet-related non-communicable diseases, especially diabetes, is a growing cancer. So it's the paper published in 2020 that Pakistan is among the top 4 countries which affected by diabetes, and the rice consumption is also high, and there is a direct link between the rice consumption and diabetes. Rice, Pakistan is currently producing 7,450 tons of rice, and on the area of large area. But however, the area is currently increasing. However, the yield is not sufficient because there is a lot of environmental pressure, and in terms of flooding, in terms of drought, in terms of high temperature and salinity. So we, as a scientist, we have to deal with climate, and we have to deal with the nutrition as well as the application of new technologies, and keeping in mind the agriculture practices in the rural areas and in the social development perspective. So Pakistan is, because Pakistan produced method of this rice to the European countries, because we have basmati rice as India, and basmati is mostly like in these countries, and non-basmati rice also we export in other countries. However, food security is still a challenge because, as the definition it says that food security exists when all people, all times, have physical, social, and economic access to sufficient, safe, and nutritious food, which meets their dietary needs and food preferences for an active and healthy life. It is not, if it is not achieved, that active and healthy life, it means the food security is still there. So what is our goal as a scientist? So our goal is to achieve zero hunger or zero hidden hunger by 2030. That requires new and existing applications of science and technology innovations across the food system, and we must address all the dimensions of food security. And innovative capabilities are critical not only for ensuring nutritious food all time, but also for harnessing agriculture and broader food system as a driver of economic and sustainable development. So food security is usually framed into four dimensions. One is food availability in the field. So most of the efforts of scientists is to ensure the availability of food in the field. And secondly, food access, which is from field to the end users. And third is food utilization, means if the food which is available in the field and which is access for the people, is it able to utilize or not? And the third dimension is food stability. So we must address all these dimensions if we are working on it as a food security, because food availability is linked with climate resilience, and food utilization linked with healthy rise. So I will just briefly introduce you both of these two fields. So what are the problems associated, and what are we what opportunities we have to solve these issues. So food availability is to improving yield, first thing, and secondly to improving input, and third is the improving monitoring system, the crop management system. Because why we need to improve the yield? Because there are stresses, there are pressures in terms of biotic and abiotic factors. Biotic means in which any biological organisms are involved, like diseases, virus, fungi, and etc. And abiotic means the high temperature, salinity, drought, and all these kinds of environmental stresses which hampers the yield of the crop. Either the crop itself is low yield. So both these two challenges we have in the field condition. Secondly, the improving input means if, for example, if the soil is low fertile, so we need to improve the soil fertility. And if there is lack of water, because water shortage is a huge issue in developing countries like Pakistan, in other areas, because in Pakistan, some rice cultivation in some areas is banned due to the lack of water. And improving monitoring system means scheduling inputs. We need to schedule your inputs on time management level, and power and control system. So how we can address all these issues for one by one. For example, biotic and abiotic stress, we need a stress tolerant crops. Means that the crop would be able to produce more food under conditions of unfavorable conditions. So low yield means we need to improve the yield of that variety. If the soil is low fertile, we need to improve the fertility by using different approaches, by using different biological approaches. And if the water is unavailable for arable lands, then we must work on the water use efficiency of that crop. It's water utilizing technologies. And for scheduling, for improving the monitoring system, we must take the technology for imaging and farm management system and robotic technologies. We all have these options, these opportunities to address all these issues. So my focus is to stress tolerant crops because we have, I mentioned that we have environmental stresses in the field and as well as lack of water. And because Pakistan is among the top 10 most affected countries due to high temperature, and the regions, most of the rice crop growing regions are already experiencing high temperature, and so therefore because rice is important staple food crops of Pakistan, so we have need to work really on identifying and exploring the mechanism, the science behind high temperature stress tolerance, and we are really working on that. But still, we need to do more work on that level. And food utilization have two categories: either the food is lack of nutrition food, or they have other non-essential elements like heavy metals, arsenic and cadmium. So if the food is lack of nutrition, for example, there are available vitamin A enriched cassava, maize and potato, iron and zinc fortified rice, and high nutrient stable crops we can make through scientific approaches. And for to mitigate the to mitigate the heavy metals, we need low arsenic and low cadmium rice, particularly focusing on the rice. So iron and zinc fortified rice we need, because as for rice is that lack of their, we eat the polished rice, which is only based on the rice grain starch, which is considered for 90% of the grain weight of the rice grain is starch. So there is not much for to eat rice as a food. And secondly, there is reports are available that rise due to the human activities, the arsenic and cadmium level is increasing in food crops, or because they are linked with numerous issues like cancers and like others. So we really need to work to improve the food utilization by developing healthy rice. So as I told you that rice grain is mainly composed of starch, which is constitutes 94%. Rice starch itself is composed of two polymers, amylose and amylopectin, which determines the quality of that rice. And amylose portion in rice starch is more important because as compared to amylopectin, amylose is more healthy in terms of low digestion, in terms of low glycemic because when you eat rice at a high quantity, if there is not much amylose and the rice is more composed amylopectin, it means that the digestive system will convert all the starch into glucose, and your blood glucose will suddenly change, and it will really affect your blood glucose, and it is very dangerous for diabetic patients and other obese patients. So, and secondly, as I mentioned that arsenic and cadmium also coming out from the ground water in food crops, both in aerobic and in anaerobic conditions. So we really need to mitigate and make the strategies to improve all these issues in a scientific way. So our food system itself in a crisis. So the malnutrition, biodiversity loss, climate change, and environmental degradation challenges. All these challenges are interconnected. So we are working on the nexus of agriculture, nutrition, and environment. So our solution must be interconnected. So integrative, recognizing energies, synergies, and trade-offs. So biotechnology is a significant contributor to these solutions at the nexus. So what are the solutions? The solution is to design and strengthening an agricultural innovation system, which involves promoting research and development and investing in infrastructures, building human capacity, and strengthening knowledge flows, particularly among scientists and farmers, and regional and international collaborations. So all these points are really important to make agriculture innovation system and to address these important issues at humanity level, because really the people in developing countries and in Asia, in Africa, they only have limited options for food, but the food itself is in challenge because there is not much for their nutrition, for their healthy life. So agriculture biotechnology. In agriculture biotechnology, we can improve rice yield by two options: either the genetic improvement or either cultural practices. And genetic improvement, you know that conventional plant breeding, we have either molecular and biotechnological approaches. And molecular and biotechnological approaches are divided into two parts, which is marker assisted selection or genetic transformation. So here is the way, two ways you have to go. You can choose the way here as a biotechnological approach, but the limitation is that just like in Pakistan, because as you know that our rice, most of the rice we export in European countries, and there is genetic transformation, transgenic rice is banned in European countries. So we currently are not using any transgenic practices in rice for improvement. For rice improvement, we are not applying genetic engineering because due to national interest. So we have the options of marker assisted selection, which means that you explore those varieties which have the ability to produce more food in times of climate change, as well as they are more nutritious. By using different markers, by using markers means from physical morphological markers to DNA markers. So we are using biochemical markers like enzymes, like proteins, and like DNA markers. So we have completed our high temperature stress tolerance study in Pakistan in here, and we reported the marker like the protein markers, how their expression level changes from time to time. So those varieties which have better expression system of those markers, they have strong expression system, they have the ability to produce more food under high temperature stress. But the varieties which have low expression of these proteins, they are more susceptible towards high temperature stress. And we are currently working on nutritional aspects. Currently, our project is going on about the grain nutritional characteristics like starch characteristics. So in which we are working on to explore the glycemic index of Pakistani rice varieties, how they're affecting the blood glucose level, and their responsible genes, like they are well known that most of the genes are responsible for a polymer of amylopectin and the other enzymes are responsible for amylose. So how we can balance these two, though that will help the obese people or the diabetic patients. They can also have rice in their diet, and the other pupils which can who can use those varieties as a healthy rice. So we hope that in future we will really do some applicable work for the benefit of human being, for the benefit of our people in Pakistan, and of course for the humanity all around the world. Thank you so much from my side. So if you have any question, you can ask.
Wow, doctor, very well done. What a yes, very nice presentation, very powerful presentation. So I would like to invite our next speaker. Please stay tuned with us, audiences. We have a panel discussion after the end of all the speakers talk, and we will have our questions and answer sessions at the end of that all these talks. And so we move ahead. Thank you so much, Dr. Muhammad K. Ali. And we have our next speaker, Dr. Naveen Sultan from Agricultural Research Center, Suez Canal University, Egypt. Dr. Naveen Sultan, are you here? Dr. Naveen? Dr. Naveen Sultan?
Move on, Shalini, to the next speaker.
Okay, no problem. So
Maybe she can join at the end.
yeah, sure, no problem. So we have our next speaker in the series is Dr. Dalshad Aziz Darwish. Dr. Dalshad, I hope he's here. I saw him.
I'm hearing you.
Yes, perfect, wonderful. Thank you so much
Thank
being
you very much.
here on time. So Dr. Dalshad Aziz Darwish is from Salahuddin University, Sciences College, Environmental Science and Health Department. Um, and he would be talking on a very interesting topic, that is health risk assessment of some heavy metals in different legume seeds brand which is imported to Erbil city. Let me briefly introduce to Dr. Salahuddin, Dr. Dalshad. So he has graduated from the Department of Biology, College of Education, University of Salahuddin in 1993 and 94. And later he joined Department of Biology as assistant researcher in 1995. Later he successfully completed his master degree, especially in plant nutrition, and then that vary from Department of Biology, College of Science. And then later on he did PhD in Department of Soil and Water Agricultural College in the same university. And he has been working as an academic in the Department of Sciences and Environmental Health Science College, and studying both graduate and postgraduate students in the very subject of soil science and soil pollution and ecostatics. And in general, his research interest is in the area of study of soil pollution, trace elements, plant nutrition, remediation, biostatics, environmental indices, plant ecophysiology, and he has been supervising many students for their higher studies. He has been helping the students for getting into expert in this very area, and apart from that he has been actively engaged in publishing various research papers in a journals of high standards and high repute. And we are very thankful, Doctor, for being here today with us and sharing your expertise in the very interesting subject area. So thank you so much, and platform is yours.
Thank you very much, Nadi teacher, for nice introduction. First of all, I would like to thank University of Duhok and Women in Science Without Border initiative to give me opportunity to participate in this kind of scientific meeting by a research title under health risk assessment of some heavy metals in different legume seed brand imported to Erbil city. So, give you a brief introduction about my article. As you know, legume belong to Leguminaceae or Papilionaceae family. Legume seeds are rich in source of vitamin, minerals, and fiber, and also have a beneficial antioxidant effect. But one problem that related to accumulation of heavy metal in soil and plant is of increasing concern because of the potential human health risk. Heavy metal contamination of food item is one of the most important aspects that related to food quality assurance. So heavy metal accumulation in plant, as you know, depended upon plant species and the efficiency of different plant in absorbing metal that calculated, evaluated by either plant uptake from the soil or from soil to plant transfer factor of these metal. So one of the most important pathway for moving these metal or toxic metal pollutant from the soil to human is via plant nutrition or plant content. Excess accumulation of heavy metal in agriculture soil through both natural and anthropogenic activity may not only result in soil contamination, but also lead to elevated heavy metal uptake by legumes, by plant, particularly by legumes, and thus affect food quality and safety. The problem of behind to select this title is a huge quantity or large quantity of legumes from different countries imported to Kurdistan region without analysis and quality control testing, unfortunately. Thus, this study aimed to assess the health risk of some heavy metals in different brands of legume seed imported to urban city. Various legume seeds, lentil, bean, chickpea, for very common or 5 very common brands were selected randomly from a market of herbal city. The experimental design, designed it in CRD, completely randomized design with replication. That means the one-way classification of data. After collecting the samples grinding by using stainless steel grinder, oven dry, then the sample digested by the mixture of H2O2 and H2O4 acid, 1 volume by volume. The digested sample were used to determine the concentration of manganese, zinc, Ag, cadmium, and Pb by atomic absorption, flame emission spectrophotometry. After determination of this heavy metal or toxic metal, the health risk assessment were calculated by use equation as described by Kane et al. 2014. The first equation referred to average daily intake, ADA, in some references right by EDI. So the average daily intake is equal to CI. The CI here is a concentration of heavy metal that expressed by milligram per kilogram. IR, average daily consumption. According to our survey, the average daily consumption by human or adult person is about 350 grams per day per person. The BW capital is body weight. Also during our survey, the body weight of adult person is about 68 kilogram in our study area. The second equation that used for calculated the hazard or health risk of this element, hazard quotient, which is symbolized by HQ, equal to average daily intake divided by reference dose or oral reference dose that established by EPA, European EPA, Environmental Protection Agency. Each metals have its own oral reference doses. After calculation, the hazard quotient, the hazard index is the final indexes, which equal to summation of the hazard quotient of each metal. So the value of hazard quotient and hazard index, when smaller than 1, that means there is no potential health effect. While when it's greater than 1, that means there are a potential health risk due to exposure for a frequency of time. So the main result that obtained from this study, as shown in table 1, concentration of heavy metal in legume seed, the variation or significant variation were found between legume seed. The level ranged, or the level of manganese ranged from 32.76 to 76.11, while for zinc ranged from 25.39 to 68.73, and for Ag from 0 to 5.78, and cadmium from 0.12 to 10.58, and for PB from 9.12 to 27.86. According to this result, the level of manganese, Ag, cadmium, and Pb is in all legume seed greater than the WHO recommendation level of contamination in food product, except the zinc concentration. Table to show the concentration of heavy metal in different legume brand. The concentration of or the data analysis in this kind of table show no significant variation among the legume brand for all heavy metal except the cadmium, which recorded as significant differences between the legume brand. So this variation between or among legume brands or legume species or genus may be attributed to the soil property, or maybe attribute to the land management or fertilizer use, or maybe related to the kind of irrigated water that used for irrigation of this kind of legume, or maybe related to genetic and physiological variation that occur or present between legume brand and also the legume species. The result in figure 1 shows hazard quotient value of some heavy metal in different legume seeds. The result indicates the hazard quotient for PB is greater than 1 for all legume genus or species, while cadmium, the higher level of hazard quotient recorded for cadmium in bean and chickpea, while for Ag recorded from bean. So according to this information or result, the lentil, bean, and chickpea maybe for some heavy metal hazard index is unsuitable or unsafe for human consumption. The result in this figure, that is figure 2, show hazard index value of different legume seeds support the previous result, which show a higher hazard quotient index or hazard index for lentil, bean, and chickpea. So according to this or these indices or hazard indexes, the lentil, bean, chickpea are unsafe for human consumption due to potential health effect. Figure 3 show hazard quotient value of some heavy metals in common legume brand. According to the result that we obtained from our study in this area, the hazard quotient for PB in all legume brands are greater or were greater than 1, while for cadmium greater in Aspirash brand and Norus Altunsa. For Ag is greater than 1 in Norus and for Altunsa, while for zinc and manganese is smaller than 1. So we can say that this brand that imported for our region, unfortunately, is very unsafe for human and for food or for human consumption. So our conclusion in that obtained in this study, determination of heavy metal concentration in legume and food product is important for health risk assessment during food consumption. So the result indicate that according to average daily intake, hazard quotient, hazard index value, all tested legumes were highly contaminated with heavy metals. Thus, the researcher tend to suggest that the legume from market under the study unsafe for consumer due to their frequent consumption, and I hope this kind of or quality control of our country be careful about this kind of food that imported from the different country to our region. This study not concentrated on every, because this brand and this legume is very common brand that distributed in Iraq overall, not in Kurdistan region only or Erbil city alone. Thank you very much for your attention.
Wow, thank you, Professor Dalshat, for such a wonderful overview on heavy metals. It's so interesting. Already, our audiences have questions about it. Dear audiences and friends, we will have a live question answer session at the end of all the talks. Please keep all your questions immediately written somewhere so that you do not forget, for our panelists, for our speakers, so that you can post them to me directly or in this chat box. So we will move ahead with our next session. Thank you so much, Dr. Dalshat,
Thank you very much.
for being here today. See you in some more time. So
Thank you.
we'll move ahead and we have our next presenter, Dr. Naveen Sultan. I hope Dr. Naveen is here with us. Dr. Naveen? Oh, okay. Dr. Naveen is still is not able to join. So we will go ahead with another
But Dr.
speaker.
Naveen is on the list of attendees. Why
Okay,
she is not clear here?
able to, so she has to be get converted from attendees to
Okay, just continue.
panelists.
And
Yes,
just
okay.
continue. Yeah.
Okay, thank you, Amal. We will move ahead, friends. Sorry for the technical glitch, but we have our next speaker in this series is Dr. Duha A. Kadim. And Dr. Duha A. Kadim is from sorry for my pronunciation if I do not pronounce it correctly, but I will try. Mustan Syria University, Iraq. Dr. Duha will be talking on iron oxide nanoparticle synthesis using trigonella and tomato extracts and their antibacterial activity. Dr. Duha Kadim has been working and she has got her MSc degree in physics science from Al Mustansiriya University since 2020, and she was also awarded the first place in the master's degree with an average of 87 percentile, and she has also got an honors of excellent degree with a degree of 94% in her thesis. And she has been working specializing in the research related to the topic of very important topic, that is sustainable development and green synthesis. Sustainable development goals and green synthesis has been a very recent topics which is gaining much importance at the national and international levels due to its significance in research university with a certificate of appreciation and excellence in the research. She has been publishing so many research papers, and we are very thankful, Doctor, being here today with us, interesting topic. So thank you so much, and over to you, Dr. Duha.
Hello, everyone.
Yes, you're very
I
much.
hope that you get my sound.
Yes.
Thank you, Dr. Silani. It is my honor to be here with you, everyone. Good evening, everybody. It is my pleasure to be here with you all. My name is Duha Abdel Karim Kadum. I am from Al Mustansariya University from Baghdad, Iraq. At the first, I would like to express my kind thanks to the University of Utrecht and the Women in Science Without Border initiative. I will share now my presentation with you. I hope it is okay with you all. Is this?
It's not yet visible, Dr. Duha. I hope you have to share. It will come because it is showing that screen is sharing.
My presentation now is, can you see it?
It will come. You'll have
Just
to
a moment, please.
Yeah, do it again.
Now, can you see my screen?
Yes, the screen, but I think it is mobile.
It's okay. Yeah, it is a mobile.
Yeah,
I
sure,
use it.
no problem.
Now,
Yes.
can you see my? It's okay. Thank you all. Thank you, Dr. Sholani. I would like to send my regards to everyone here. Listen to my voice. And of course, to the University of York and Women in Science Without Border Initiative. So I would like to be focused on my research entitled of iron oxide nanoparticles using trigonella and tomato extract and their antibacterial activity application. And here we go. I will divide my presentation into three main parts. The first of all, I would like to talk about the outline of this research and the abstract. The second, I will look at the preparing of the nanoparticles and how we make it. And finally, we will explain the result we got and the conclusion in this research. As you can see with me, this is the abstract of our research. So let us start with the abstract. Iron oxide nanoparticles can be prepared by combining the extract of two planets, Trigonella and tomato, with ferric Also, we will show that in the result part. As you can see in this picture, this is the trigonella and FeSCl3. Then we will applicate it in antibacterial application. Here we go. We will move to the introduction. We will prepare iron oxide nanoparticles in a modern materials by using biosanitized method. Is a developing field in nanotechnology. It have various important application in various fields such as biological, chemical, physics, biochemical, biomedical application. Iron oxide, we will move to iron oxide. I will be short talking about every part to be much with the time we have. So iron oxide is in this work, we will talk about iron oxide specifically. Iron oxide is found naturally and easily to manufacture in laboratories. We here talk about the green methods is eco-friendly and Okay. We here talk about the iron oxide and sanitized by the green method. It is eco-friendly and eco-chemical and costless in contrast with its scientific application fields. So we will talk about the next step in this work and experimental work, FCL3. We will take 16.2 grams of FCL3 and 100 ml of water, distilled water we use in the laboratory. Plus we will add it to 20 ml of 20,000 ml. 2,000 ml of trigonella to preparing 1 molar of mixture. The solution will be produced was magnetically stirred at 80 degrees Celsius for 30 minutes in the laboratory. the color of the mixture will change, as you can see in this picture C. After we mix picture A with picture B, then we will move to picture C. Picture C show that it has suddenly changed the color of the mixture to black by mixing to solution we use. Sorry for this interruption. Then after we cooling the solution gradually to the temperature of the laboratory, we will get a small amount of the solution. Then we will put it in ceramic dish and heat it and vaporize it for 2000 degrees Celsius for 2 hours to get the nano powder, as you can see in picture D. Picture D shows that this is the iron oxide nanoparticles powder we get after this procedure. Then we will move to the next part of our research. It is the part of result and discussion. As you can see in this picture, it is show that
Please, doctor, we see no pictures. You have
there is no
to
picture
move
you
on.
can see?
Yeah, no. You
I'm
have to
sorry
move
for
on
that.
with slides. There is a problem.
I will
Just move
share it
on.
again. I'm sorry for that. I hope.
I think only the slides, if you would have done next, it would have been seen. No problem.
Did you see now?
Yes, we see the table.
I hope it is obvious for you all.
Yes.
Good. In this picture, we see the XRD peaks showing that the iron oxide, we always take the XRD measurement to make obvious that the crystallite size of our mixture or our nanoparticles we got. So in these pictures, there is many peaks are obvious for the 3 different solution we got. Crystallite size is about 17, as I put it in this table. Is this obvious for you? Is my
Yes,
screen?
yes.
Yeah, good.
Go ahead.
Good. So in this picture, I put it all. So the crystallite size is about 17 nanometer for the iron oxide nanoparticles made using trigonal extract with the FeCl3. And the crystallite size is about 41 nanometer for iron oxide nanoparticles made using tomato extract, as you can see in this. So, and the Miller indicate show that it is given a body center cubic phase. Believe that it is a body center cubic structure phase. Then we will move to the next structural measurement, the FESEM analysis of the iron oxide nanoparticles using trigonal and tomato extract. In the first picture, A, we will see that it is a picture of demonstrate the shape of the nanograin structure observed using a trigonal extract. This is for trigonal extract. Okay, so it is shown that the planet extract for trigonal given about so in the FESEM analysis, we will focus on the average grain size we got. Average grain size. So in the trigonal extract was 28 to 41 nanometer. It means that it is very small particles we got to 41 nanometer. And in the next picture, picture A1, as you can see, this picture A1, we will talk about the tomato extract, which can be measurement. The average grain size of it is 40
Hello?
Yes, continue, please.
Yeah, Dr. Doha, your slide is not
Oh,
moving.
she left. Oh, okay. Oh, I thought this is call from her. Okay. We might wait for her maybe within 1 minute.
Because I think her slide is seen, but
Yeah,
it's
she
I
not.
think
missed the
I think she has problem with her computer.
computer, or maybe even she might lost the connection.
So that we can transfer after 1 minute to the next speaker, Dr. Niveen.
Yeah.
She is not there in the meeting, but this presentation needs to be stopped. Then only the other presenter will be able to present.
Let me check if I can stop her presentation. I can stop her sharing. So let's wait. When she's not available, then I can do it.
That's fantastic.
Yep. Yeah.
I still see the slides. Or are you going to wait for some more time?
No, I think we have to move to Dr. Niveen.
Yes, already we are.
Dr. Naveen is already not there. Yeah, she might miss this one. She's absent.
No, she's here, but she
Already?
couldn't open
Niveen,
the
yes.
video or audio. I think she has problem as well.
We will see.
Not.
But can you stop this sharing?
Yes, sure. Stop sharing. Yeah, I stopped.
No, I see the slide.
This will stop other screening. Do you want to continue? Yes. Let me just try to stop hers, then I can simply stop mine. Yes. And I can stop mine. Here you. Oh, she again.
I still see the slide.
She left. She left, and her slides here.
You know, I have the I mean by right click on her, I can see I mean I have the option stop sharing, so I should stop. Oh, she's back. I think she's back with 3 accounts. You may please enter the session using one single account. Yes. Let me check.
Can you hear me?
Yes,
Yes.
Dr. Doha. But please wrap up.
I'm sorry for that.
Please wrap up your lecture. Come to
Yes.
conclusion, please.
I will continue and finish
Only
it all.
conclusion, please. Come to
Just I need a
conclusion.
confirmation. Does Dr. Naveen's name is Naveen Abdullah Hamid?
Yes, I think.
Okay, then okay.
Can I complete it now?
Please wrap up this. Wrap up it.
Yeah, but just do it quickly, please.
Very
Okay, here
quickly.
we go. It is the FTIR, so that the iron oxide nanoparticles, and give it the band of it. So it is obviously it is there. The FeO, it is there, and it is band showing. So we will talk about the important part of our research. It is the antibacterial activity application. So do you get me?
Yes.
Did you get my voice? Okay.
Yeah.
It is
Just continue.
the antibacterial application. The first one, as you can see, it is Klebsiella pneumonia, gram negative, and the second picture is Staphylococcus aureus, gram positive. As you can see, the Klebsiella pneumonia negative gram gives the highest inhabitation zone for the iron oxide nanoparticles prepared. So as I show here in this table, this is the percentage of habitations. Then we will move to the conclusion. Our conclusion is that the iron oxide nanoparticles sanitized using tomato and Trigonella plants, it is a real method with a good result. So it is a straightforward, advanced, and costless method to make the most important feature of it is environmentally friendly method. This is the most advantage of this method. We use it. And we can see in the HRD analysis, given the crystallite size about 41 nanometer for the iron oxide nanoparticles prepared. So in the FESEM, we show that our average grain size of the iron oxide nanoparticles prepared by the two extract we use, which was about 25 to 41 nanometer for Trigonella, and for tomato extract up to 77 nanometer, which is the most good advantage for this measurement, that we can be assured it is nanoparticles up to the average grain size we got it. And the last part of the conclusion, we will talk about the antibacterial activity application, and it is the most important part. The antibacterial application for Staphylococcus aureus and Klebsiella pneumonia show that it is work very good for the gram-positive bacteria and the negative bacteria, and it may be effective in the healing infection causing by multiple types of bacteria, of this bacteria specifically. So we will move. I have many research publishing, as you can see in Elsevier magazine, and you can see this is our research now, iron oxide nanoparticles, publishing in Materials Technology, and this is our international conference. It is all about the biomedical application. And at the last, I will thank you everyone for your attention, and thanks for Al-Mustansiriya University and its laboratories. Thank you all. Thank you, Dr. Shoilani. And I'm sorry for the multiple corruption that have been, it is out of my control. Thank you all, and thank you, Dr. Shoilani. Thank you. Thank you to all participants here.
Thank you, Dr. Doha. If you can stop sharing the screen, that will be nice.
I will stop it. Thank you very much.
Thank you so much. The latest slide that you showed, we couldn't see those, like especially you said you have publications published with Elsevier, but that slide was not moving. Only I had those two slides in front of me. Those two petri dishes with the microorganism were in front of me. Okay, so we will move ahead. We have our next presenter, who is Dr. Naveen, and Dr. Naveen is here with us. Let me introduce Dr. Naveen. Dr. Nevin is currently working with Agricultural Research Center, Suez Canal University, Egypt. And she would be talking to us on the topic of mites inhabiting some fruit trees in Egypt. Dr. Nevin has received her PhD in ecological studies, especially entomology and invertebrate structure, from Suez Canal University, Islamia Government, Egypt. Her research interests are focused on especially biology, biodiversity, agriculture studies, and sustainability. And she has also done PhD in ecological sciences in 2018, wherein she has determined the biological diversity of the fauna to contribute onto the conservation trend of different animals and insects, especially in the Egyptian environment. And she has been working as a researcher in an Egyptian research center, and she has published multidisciplinary research publications belonging to the ecology, biology, and archaeology, biological control sciences, and she has been the first author of these publications. So Dr. Nevin, we are very thankful to you for being here today and sharing your research work. Over to you, Dr. Nevin. Yes, Dr. Nevin. Dr. Nevin? Hello? Dr. Nevin, unmute yourself. Yes, we can hear you.
No, it's me. Dr. Nevin.
Yes, I see her.
Yeah, even me, I see her. Even her mic is open, but we cannot hear her voice. If I would ask you, Dr. Nevin, if you have the Facebook open, then please close your browser so we make sure that there will be no echo between the Zoom session and the live stream on Facebook session.
I think she is with us because she's moving the presentation.
We can see her screen, but I cannot hear her voice at all.
She's playing with the screen, but no voice. Dr. Nevin,
would you please write if you are talking but we are not hearing you?
I think she has problem. Yeah. Maybe Shalini, you can run questions, and when Dr. Nevin will be ready, she will join us because of the time.
Yeah, yes. Sure.
Okay.
What you will have to do, you join again, and while joining, you have to say input audio. So there is some problem with your audio system that you have to activate while joining the Zoom. Call in over, that option comes when you join. So maybe you can try quickly that, but it should be an automatic option. Okay, so friends, if you can stop sharing, Dr. Nevin, or you want to try again, try unmuting yourself and try talking to us. We can see all your screen, your computer, your inbox, what you are doing, everything.
Dr. Nevin, we see all your computer, your screens. It's not a problem of screen. It's a problem of voice.
Yeah, so maybe quickly you get out of the meeting, and when you are joining, the option in Zoom, it comes, call in over audio, video. So you have to click on that call in over option.
Okay, that's why, Shalini, I told you go on in questions.
Yes. Yeah, Dr. Nevin, would you like to try? Say hello to us. We will give a last try.
Hello.
Yes. I think there is an audio problem. Okay, so friends, we are going to move ahead, and we have our questions for our panels. Okay, so all the audiences here can post their questions here in the chat box directly to me or in the chat box. Okay, and Amal, would you pull out the question from the Facebook live session there?
I think no questions on Facebook.
Okay, so
Only
I
watching.
have few questions to ask Dr. Mohammed Ali. Your presentation was very interesting, and few questions that I have received. Based on that, I would like to ask you, have tried to use the gene technology or modification of gene, and especially to modify the rice to be consumed by the diabetic population. And you also mentioned that you target basically the amylose and amylopectin constituent of the rice. So according to you, which rice is good for the consumption? Whether it is sticky rice that is generally consumed in some of the Asian countries like China, Japan, or the rice which is non-sticky. In India, it is very famous. We make, and even in Pakistan, we make that pulao or rice, which is like not non-sticky. So what are the gene characteristics or speciality that leads to these two kind of rices that are generally consumed, and what is its correlation with the diabetes?
Oh,
Yes.
thank you so much. It's really a nice question. You know that on the basis of grain starch quality, there are two types of rice, as you described. That one is sticky rice, and other is firm rice, which is not sticky. And we in India and Pakistan, we eat more non-sticky rice, which is and Japanese and Chinese use sticky. Right? So for the diabetic patient, non-sticky rice is good as compared to sticky rice, because it was sticky in terms of waxiness. If there is much waxy, that the rice is sticky, it means the amylose content is low as compared to amylopectin, right? So in starch, these two polymers determines the quality of rice grain. If there is high amylose, the rice is non-sticky. And because amylose is not branched, it is a linear polymer. So the enzyme in our digestive system, amylase, cannot break easily. You know, the alpha amylase in our digestive system, from the mood to the small intestine, there's enzyme which break the starch into glucose. So if the enzyme can easily break amylopectin, which is highly branched, as compared to amylose, right? So if the portion of the rice grain, if the portion of the amylose is high in the rice grain, that is non-sticky. So the non-sticky is good for the diabetic patient as compared to the sticky one. But there is another factor, for example, the chain length, how the polymer is a very large chain or there is a small chain. Because if you cook the rice, during cooking, the small chain leach out in the water and you drain the water, But the large chain again polymerize and make the crystal again. So if the chain length of the polymer is long, it is good for diabetic patients. If the chain is length is small, it means the starch is not good for diabetic patients. So in conclusion, simply that non-sticky rice is good for diabetic patients.
Oh, thank you so much, Dr. Mohammed. So, amylopectin is a long chain polymer. So, do you think that the long chain, because amylopectin is a long chain polymer, and interestingly, if you see, although we consume a high amylose containing rice in India and Pakistan, unfortunately, India is amongst the topmost country affected with diabetes.
Yes.
So looks like very contradictory. And what is your comment or opinion on that?
Yeah, in the sense that the rice we eat more, we must reduce the portion of the rice we eat. Because if we eat a food very high in amount, it affects, of course, on the blood glucose level. And other cooking characteristics like their gel consistency, gelatinization, but there are other factors that involve that which makes the rice is more glycemic. But as you know that it's simply in the word that if you eat 2 times a day a rice, so it of course affect your blood glucose level, right?
Wow, excellent. Dr. Muhammad, your topic is so interesting. I will come back to you yet with another question, but we have a question for Dr. Dalshad, who has worked on heavy metals. I hope Dr. Dalshad is here. Yes,
I
Dr.
hear you.
Dalshad.
I hearing you.
Yeah. So you mentioned a very hard fact, indigestible fact, that the legumes are considered as a protein, and especially for vegetarian people. At least in India, we have a lot of vegetarian people, and across the world, I've seen some of the countries who are, talking and or adopting vegan diet, where the legumes are becoming boon as a very high source of protein. And you mentioned that these legumes, if the farmers do not practice a correct farming practices, the heavy metals can cause, they could be very, very deadliest. So, as a common person or as a common consumer, what can be done at a home scale level so that at least a little load of this heavy metal could be reduced, and the health benefits of these protein could be enjoyed and released upon? Yes, Dr. Dalshat.
So the nutrition of the legume is very important. As you say, the protein is very essential or very primary and original property or as a chemical composition. But these plants able to uptake the heavy metal from the soil that polluted by these heavy metal. Unfortunately, maybe in some country uses wastewater for irrigation. So this heavy metal reach the soil and enter the plant and transfer to human body. So I think it is very good for consumer. But we remember in our article that the heavy metal is the second dangerous or the second dangerous poisonous or toxic metal or toxic material that reach the human body via the plant. So we can say the nutritional quality, which is involved with the protein, carbohydrate, or vitamin, and so the metal nutrient are very interested here. But our work is concentrated on the all brand of legume that imported to different country to our region in Kurdistan region, Iraq, is very huge quantity without controlling quality, without analysis for this heavy metal. Our solution is, or our study focuses on the hidden hunger of this element that is very toxic for human health or for toxic for environmental health. So instead of thinking about the chemical composition, the nutritional composition, which will involve with the protein, so it is very important to concentrate it on this metal. This metal is a toxin, and frequently, as you know, frequently used by people for consumption. So accumulation of this heavy metal maybe causes cancer, as a carcinogenic agent, and maybe affect the nervous system.
So, Dr. Dalshad, sorry for interruption. So, do you think that, because heavy metal identification is not a single task and cannot be done by the normal population, so do you think the government organization or those bodies who takes care of food safety and standardization respective to each country should look upon such heavy metal content? That is one thing. That's what I have understood from your talk. Am I correct?
No, as you know, the pollutant is very variable. There is a different kind of pollutant that come from this food to all country, like the polyaromatic hydrocarbon, as we say, the heavy metal, and so some pesticide that absorbed by plant may be accumulated inside the plant as organic composition. So this material is very dangerous that come from food to human. In some situation, we can say come from airborne or come from water to other country, but when come from food, as you know, the importance of this food is very huge. Maybe come from the border country or from the other country to our region. So the government or governor in general must be focused on this point that related to heavy metal toxic. So I think it's very interesting article about this pollutant that come from food to any area around the world.
Wow, that's very eye-opening, Dr. Dalshad. I would get back to you with another question. I have a next. Thank you so much, Dr. Dalshad. I'll get back to you with another question later on. So right now I have a question for Dr. Duha. So Dr. Duha, are you there?
Yes, I'm here.
Yes, Dr. Duha, very interesting presentation you showed on tomato extract and trigonella extract. So tomatoes are part of our every delicacies, and in some of the countries, the tomatoes are eaten raw. Tomatoes are rich in abundant of vitamin A, then lycopene, and so many other bioactive constituents. So you have worked on these nanoparticle in nanoparticle making of these extracts. So what is the impact on bioavailability of these bioactive constituents?
Impact what? So would you repeat after impact?
Yes, it's when you consume. Suppose I consume tomato as such raw, and the one that is encapsulated or nano capsulated, nano converted by you. So which would give more activity compared to the raw one?
Okay, I didn't get your question well. So as I understand, you mean why tomato extract and why trigonella? Did you mean that? And why we use it in antibacterial activity?
Actually, I mean to say you have done a nanoparticleization, right? You have made nanoparticles from tomato extract, and
yeah.
Right? So what is its impact on digestibility or bioavailability? So it is for human consumption, right?
Okay, yeah.
How it would when I consume the nanoparticulated extract over the fresh tomato puree or fresh tomato, the bioactive that I'm going to get through the fresh extract and the one through the nanoparticulated, which one would be more beneficial giving the activity?
The trigonella was the most activity in with the antibacterial activity, because I think it is of concentration of iron oxide inside the trigonella is most in contrast with the tomato. Tomato is rich with vitamins, rich with many materials and many metals is needed in the human body, but the trigonella, it is a vegetable, so it is more rich with metals as iron oxide. So it is given most bigger and the most effective, as we can see, most effective inhibition zones. It is good for this application.
Wow, excellent. Thank you so much, Dr. Duha. And I will again, if there is a time, we will have again few more questions. But Dr. Nevin
Thank
is
you.
with us. Thank you so much, Dr. Dalcha. Thank you, Dr. Duha. Thank you, Dr. Mohammed, for such a wonderful answers on very relevant to this topic. And we have Dr. Nevin. Can we hear you, Dr. Nevin? Oh, Dr. Nevin, we are not able to hear you. I am extremely sorry for the technical glitch that you are facing right now. I request the panelists, those who have finished speaking, do not leave. We are going to have a very nice photo session before we begin the next plenary session. So stay here for the photo session. Dr. Nevin?
She's not ready.
We are not able to hear you, Dr. Nevin.
Still has a problem or دكتورة نيفين اسمعينا. Okay, you heard us but we cannot hear you. احنا مش سامعينك. عندك مشكلة في المايك. You have a problem in the mic. We cannot hear you.
But you know, in between there is some disturbance of the noise. I think there is some problem with her. If you are using external mic, please try to remove them aside and try to use the mic.
The microphone? No, this is just test the mic.
Yes, test the microphone.
Test the mic.
Yeah, try to speak and I will see if it is working perfectly. Try to speak.
Are you
Yeah,
making
it is.
test the mic? I think there is a
Yeah,
problem with the mic.
yes, you could, because when you do the test mic, click on the test mic or test speaker.
Oh no. Oh yeah.
Under microphone, yeah.
Dr. Shalini,
Yes,
I
Aman.
think if there is permanent problem, please wrap up the session.
Yes.
But take photo at first between you and the speakers because we have plenary in 10 minutes. Okay?
No problem.
Yeah, but Dr. Nivin, she might stop her share screen.
Yes,
So-
Dr. Nevin, please join us for the photo session. I request all the speakers to open up their camera and be ready for the photo session. And if the organizers can help us.
دكتور نيفين حتى لو انت مش قادره تدي المحاضره خلاص الابستراكت بتاعك حضرتك موجود في الكتاب. افتحي الكاميرا خدي صوره كسبيكر. واضح ان عندك مشكله بس في الصوت. ممكن تفتحي الكاميرا. ستوب شيرنج الصفحه. افتحي الكاميرا.
Dr. Mohammed, please open up your camera. Yes. Dr. Doha. Dr. Amil, Professor Amal, will be happy if you also join us with the photo.
Okay.
Perfect. And University of Doha, organizer.
Shaheen.
Yes.
Shaheen, open the camera.
Yes.
Yeah,
And Dr.
right.
Doha.
Let me.
Dr. Duha. So since I am not a host in this meeting, I can take only screenshot and say foods, environment, water.
Yeah, thank you.
Yeah, one more.
Here, she's there.
One more.
Thank you.
Dr. Nevin's camera is.
Thank you. Wrap up
Thank you
session.
so much.
Thanks, Shalini, for moderating this nice session. I will leave you now because we have plenary. Come with us in the plenary.
Thank you so
Bye
much.
bye.
Yeah, we
Good
also invite
afternoon.
people
Bye.
attendants to
Bye.
join us in the plenary after 10 minutes. less than.
Yes, thank you. Can somebody post the link for the plenary here so that I can join?
I will do it for you.
Yes, thank you so much. Thank you so much, Dr. Dalshad, Dr. Muhammad, Dr. Neveen, Dr. Duha, Dr. Hasim for such a wonderful talks, and I look forward in near future for more collaboration, more association, because we have a lot of similar interest in the research area, and we can definitely work on the similar line. So look forward to see you yet on another platform of such Women in Science Without Border. Thank you so much, organizers. Thank you, speakers. See you all. Bye-bye.
Bye-bye. Thank you very much.
Thank you for all.
Dr. Shaheen, can you share the link for the next session? So
Oh,
that
yes, I will share it with you in a moment. I can add
Yeah,
your
in the chat
emails
box.
to the plenary session as well.
But are you going to share it on my email or here in the chat box?
Yes, I can add your email to this session there.
Okay. Thank you so much. And I'm leaving this meeting then. I will get the email, right?